Hi everyone. Thanks all of you for registering to this webinar titled Biomedical Data Analysis using Generative AI. So first of all, I would like to welcome all of you to this webinar. And the title is basically using Generative AI. So we assume here that like majority of you must have heard of the term Chat GPT in the past few months, right? And what is Chat GPT? We know that like it's some kind of the model which has been trained over a very huge amount of the data, where we feed we give some questions in the layman terms, and it has the capacity to generate some answer and give it to you. The better you are able to give your input, the better the model is able to give the output to you, right? So in the today's webinar, which has been titled Biomedical Data Analysis, we are going to look at the application of generative AI in the field of biomedical data science, or how generative AI can make the life easier to the biomedical data scientists, students, and basically the beginners. The students who are complete beginners in the field of biological sciences, have no coding experience, but are able to understand the fundamentals of the biological sciences, and they know how to do, what to do, and why to do something, right? So generative AI is a concept which is going to make your life easier, but you need to have few domain skills, right? So second slide, Neil, please. Yeah, great. So, and this event is going to be conducted by we at TechMedBuddy in association with Dr. Neel Das, who is a senior AI scientist at Roche Healthcare, and he is the leading head of LLM-based model. So he will be introducing him in the coming slides. So it's about me. So I'm Alok, and I'm the founder of this particular company, Brass Execution Private Limited, which works with the name of TechMedBuddy and is known by this particular name, TechMedBuddy. So I'm basically a PhD scholar in the field of computational biology and AI in healthcare at IIIT Delhi. Prior to this, I have done my master's from complex system at Jawaharlal Nehru University. And prior to that, I did my bachelor's in the field of biomedical sciences from University of Delhi. So I'm someone who carries little bit understanding of biological sciences and little bit understanding of data science. So I'm someone who works at the intersection of biological sciences with the data science. And other than this, my academic career and the entrepreneurial career, I carry a teaching experience of more than 8 years. Okay. So in today's session, we are going to, you will be, we will all will be interacting and looking at the application of all these things like generative AI and biomedical science. Just one disclaimer that please do not, like, if you have any doubts in between, so you should go to the right hand side. There is a triangle button, and there you will get option, ask a question, just type your question, because it is going to be a complete hands-on session. So we'll be having less amount of the time and a lot of topics to explain. And in the end, we'll have a complete Q&A session. Okay. So over to you, Neil. Thanks.
Thank you, Alok. And it's a pleasure to be here. And I welcome everyone to this really exciting session on how generative AI could be used to accelerate your biomedical data analysis workflows. So Alok already said, so I am an tech lead who's working with large language models at Roche. So I'm currently based in Switzerland. I have done my PhD and postdoc from KU Leuven University in Belgium. And my thesis topic was machine learning for respiratory diagnostics. I've had the privilege of publishing more than 10 publications, 2 patents, and also my research attracted a lot of grants as well. In 2020, my research work was awarded the NHS UK AI Award, which is one of the most prestigious grants in the UK to further the implementation of AI in healthcare. I also worked for briefly as an AI engineer for a spin-off company that was spun off actually from our lab when I was doing my PhD. And the software that we developed there, which was again based on my research, this is now widely used in the pharmaceutical industry to do quality control of spirometry devices. And going further back, I actually graduated from IIT Kharagpur in 2011. So with that, let's begin this session. I'm really excited to talk about it because this is the topic like I work daily, and I cannot wait to like share this more with you. So before we begin, let's just touch upon a little bit on the basics of generative AI. So what actually is, and you may have already heard what Alok said, ChatGPT. I think all of us have worked with it, played around with it. So generative AI at the basics, it's a class of machine learning models or deep learning models that can generate content like text, code, images, music based on human prompts or human instructions. Uh, and these models are trained on a large amount of data to predict simply the next word. And so this is at the heart of how generative AI works. And I show you with the help of an analogy on the right, on the image on the right. So here is how an autocorrect works. For example, auto completion works. I had such a, so what would be the next word here? What is the most likely next word that's going to be here? And it will be great time most likely. So this is how, just to like dumb down on how these things work. So generative AI simply works by predicting how the next token what the next token should be. But when you scale these models to like really millions of parameters, the models become really intelligent. It's able to come up with different reasoning capabilities. Uh, Examples for generative AI include ChatGPT. I think Bard you have already heard of. Models like Stable Diffusion, which is an image generation algorithm. DALL-E, which is again another OpenAI's image generation algorithm. Uh, Today, I highly think that mastering its use is becoming indispensable because these tools can really boost human productivity. And in the market, I feel that people will have to leverage these skills to improve the output, improve the amount of output you can do. And for this, we think that there is a perfect use case for people who do biomedical research, students who are in this field. And I have often seen, and I have often interacted with people, biomedical researchers and students who have really interesting ideas, who would like to do a very interesting scientific project, but they're often limited due to a lack of coding skills, if you will. Uh, Fortunately, today we have these tools such as ChatGPT or generative AI, which can actually help students and help researchers to do their scientific projects. And this would mean that the people can re reduce the amount of time and effort doing the coding part itself. So you can actually now focus on high-level tasks such as what the research question should be and how you should design the experiments, whereas you could outsource the coding part itself to the generative AI tool. And you can do not only coding, you can also do visualization, for example. Um, you can also design like websites as well, dashboards as well. There are a lot of use cases but for biomedical data scientists or biomedical researchers and students. I think the most use case would be how you can leverage generative AI to generate code to do data analysis and also to visualize your analysis. And finally, working with these tools also offer a path to enhance your learning. Because sometimes if you want to learn a new algorithm, it's a bit difficult. There is a steep learning curve, but if you sort of work with generative AI tools, which can generate a new algorithm, you can probably learn from it and also later apply in your data analysis tasks. So I think the future is going to be that biomedical researchers, biomedical professionals who master generative AI tools, they will lead the way in solving big challenges in biology and medicine. And I think they'll simply be irreplaceable. These skill sets are really, really important in the years to come. Uh, so what are the generative AI tools out there already in the market? Of course, you have already used ChatGPT, which is free to use, very easy to use. But the issue with ChatGPT is the programming workflow is very inefficient. For example, you go to a website, you generate the code, then you copy paste the code probably in your Jupyter notebook or your R Markdown, R notebook. So going back and forth is often not efficient, Again, there are also issues of data confidentiality, which I will touch upon later in this presentation. Also, using the free version of ChatGPT means you cannot get access to the most advanced generative AI models. And finally, doing research using ChatGPT is simply not replicable. You need to biomedical data analysis demands a rigorous approach where your analysis is completely reproducible by someone else, and simply using ChatGPT does not allow you to do so. So what are the other options? What are the other alternatives out there then? Well, you can use them Copilot or such as GitHub Copilot or Colab AI, but these would cost you somewhere between $10 to $20 a month, roughly 800 to 1600 rupees. So of course it's high cost, and but then these tools actually allow you like a very excellent programming workflow. Uh, so so finally, we need a tool which is sort of a mix of both, you know. So what we have zeroed down, the best way to do your biomedical analysis, and I think personally, is to use ChatGPT APIs. And today we'll show how you can use ChatGPT APIs to develop a co-pilot type functionality using on your Jupyter notebooks, which we call Jupyter AI. And this is kind of a pay as you use, and the costs are like absolutely minimal. For example, you pay like roughly $0.0015 or 12 Indian rupees, 0.12, I think 12 paisa per 1,000 tokens, which roughly translates to 750 words. So to process around 750 words, you will just spend around like a very minuscule amount of money. And this allows you an excellent programming workflow. It's very low cost, but the only con is it requires a little bit of manual setup. But don't worry, we'll go through this today. So what will you again learn from this workshop? We'll touch upon how to set up the Jupyter notebook. So we'll actually use a use case, a use case from bioinformatics, and then using this use case, we'll show you how to set up a Jupyter notebook to use AI Copilot. We'll share some prompt engineering tips and tell you about what an AI pair programming workflow looks like. AI pair programming is simply AI and humans programming together to do their coding much faster. We'll talk about a little bit of Git basics. Maybe most of you have heard, or probably not, but it's very important to make your research shareable as well as reproducible. And for this, you should know a little bit of Git version controlling as well. And finally, I will touch upon data confidentiality. I will try to create some awareness what you should be cognizant about when you are working with this tool, especially when it comes to data confidentiality. So with this, I now hand it over to my friend Alok, who will describe the use case that we have taken up for today. Over to you, Alok. Alok, you are not audible. You're on mute, I think.
Sure, I'm really sorry. Yeah. So, hi guys. So today we are going to just look at for the demonstration purpose. We have taken a small data set from C Bioportal. So C Bioportal is a database contains the so many different modalities of the data, and from those modalities we have taken gene expression data for the today hands-on topic. Okay. So we have taken data from this particular paper, research paper, pediatric brain cancer. Okay. So we have taken a data set from the pediatric brain cancer from C Bioportal, which contains a sample size of 218. It means that there are total 218 samples and a large number of features are there. And what are the features here? Features will represent basically the genes. Okay. And what is genes? We are assuming that majority of you are biology students, you must be knowing. So gene is the fundamental unit of inheritance. And so our features are going to represent here the genes. And we have taken the gene expression count values. Okay. So in the normalized form. So the data, when we will present the data to you, then you will be able to understand that the data that we have taken for this purpose right now here is only a small chunk of the data. Okay. Which is gene expression normalized count values data, right? And the last column of the data will be basically your a condition vector. Condition vector means basically, or the clinical status, That is, we should know that these 218 samples, and the sample means say the patients. There is 218 patients belong to which particular class. And as it is a brain cancer data, so these 218 patients has been classified into the different types of the cancers, cancer subtypes. So the different categorical, the classes of the different categorical variable is medulloblastoma, ganglioglioma, low-grade glioma, ependydoma, and high-grade glioma. Then crani- craniopharyngioma and atypical teratoid rhabdoid tumor. Okay, but for the demo purpose, we have only taken two subclasses from this data. That is medulloblastoma, which will be represented by MBL, and ganglioglioma, which will be represented by GNG. Okay, fine. Next slide, Neel. So this is the overview of the data. Okay, fine. So you should know like then what is GNG and what is MBL. That is, what do we mean by medulloblastoma and what do we mean by ganglioglioma. Okay. So medulloblastoma is basically like both are the different types of the cancers. So medulloblastoma is one of the CNS, central nervous system embryonal tumors arising from the cerebellum. There are different parts of the brain, and one of that is basically your cerebellum. Okay, so it is a tumor from the cerebellum, and it is most common pediatric malignant brain tumor, and it comprises approx 15% of all the pediatric CNS tumors, which shows the impotence of this particular study of this particular cancer. Okay, that is, it is so malignant, it can lead to the death of the child and the adults as well. Okay, then we go to the ganglioglioma. So it is basically grade 1 rare benign tumor of the CNS and can occur anywhere in the CNS. Okay. So that was basically restricted to the cerebellum, which originates from the cerebellum, but it can occur anywhere in the CNS and including the cerebrum, cerebellum, thalamus, spinal cord, hypothalamus, and the other parts of the CNS subparts. Okay. So they are the two subtypes of the brain cancer that we are going to study in today's session. Okay. So what are you going to do? That is what is your objective for the today's hands-on. Let us see that. So in today's session, we are going to find out some signatures. What do we mean by signatures? So signatures means basically like some set of the genes having the potential to discriminate between the two given classes. And as we are targeting here MBL versus GNG, it means that the set of the genes that we will be finding, they should have the potential to classify MBL from GNG. It means that with the given set of the genes, by looking at the expression values, you will be able to predict that if the expression value of these genes are going into this order, then this patient has the more probability to fall under GNG class. Otherwise, MBL class. Okay, so this is the meaning of signature, and our target for the today's analysis is this: to reaching out that particular set of the genes and how we can reach there with the help of generative AI. So in order to do this particular task, we have to follow this particular methodology. Okay, that is, we have to find differentially expressed genes. Okay, so we have to find out first of all the differentially expressed genes, and as I told you that the data is already normalized form. Okay, so we can simply perform any statistical inferential test method to find either the mean of the two classes significantly differ or not. Okay, and for that purpose, we are going to perform the t-test here, simple t-test. Okay, so we'll be performing t-test to find out either there is a significant difference with the two classes or not. After that, we'll be looking at with the help of those set of the genes, how well the patients are being able to differentiate it. And we'll try to look at the distribution of the patients. Okay. And as we know that we have a very large set of the genes, that is approx 20 to 30,000. From that, we have taken only 2,000 genes for the today's hands-on. So we'll be finding some set of the genes like 50, 70, 100. Then again, a problem will come that how can you visualize those 50 dimensions? So we'll be performing some dimensionality reduction, that is PCA, and we will use PCA for the visualization purpose. After that, we will perform enrichment analysis. That is, we will try to look the set of the genes that we have identified, in which particular pathways are they enriched in. Because if I'll talk about like Fox gene, so you will say that what mad are they talking about? But if I'll tell you that I'm talking about cancer, then you will say that, oh, yes, I'm able to correlate. Okay. So enrichment is something where we are able to correlate the genes with the pathways in the body. Okay. So for that, we're performing enrichment analysis to look for the pathways the set of the genes are enriched in. And which genes? The genes that we'll find in the objective one. And after that, we will build a ML based classifier using the set of the genes obtained from the differential expression. Okay, that is, we'll try to see like how well we are able to differentiate the class 1 with the class 0. Okay, yeah. Next slide, Neil.
Okay,
Great.
thanks, Alok. Thanks for
Yeah,
explaining the case study. So now let's begin with the actual fun exercise, which
hands
is the
on.
code walkthrough, the
Sure,
hands-on exercise. So let's begin.
sure.
Let me share my screen. Uh, so, so today in this hands-on exercise, will be using Jupyter Lab. So Jupyter Lab, which is an open source free Python software. Uh, So the setup is actually based on this. So you can scan this QR code to get the actual GitHub repository link. Um, and this repository link contains instructions on how to actually set up the whole system. Uh, we have installed many packages, but the most important are Jupyter AI, OpenAI, and also OpenAI API keys. So API keys, you can think of it, these are keys which allow you to access the OpenAI models in a programmatic way. So when you are using the ChatGPT web app, you are simply interacting and chatting. But when you have to use it in a programmatic way, from your notebook or from your scripts, you will need something called an API keys to access these models. And I will quickly show you how this works. Let's go back to the our notebook. Okay. First things first is, of course, you need to set up your whole environment, so whole Jupyter Lab. Now, I have set this up on my local computer. It's a slightly more advanced way, but you can also replicate this easily on Google Colab, which is a free Google-hosted Jupyter Lab. You can go to googlecolab.com. I think I have included the links in the repository. You can see this is our repository where all the instructions are mentioned. You can see how you can run this whole notebook on your Google Colab as well, which is much easier to use than what I have shown. But on this, the reason why we are not using Google Colab today is because the Jupyter AI functionality, when we were testing it was not working properly, and hopefully Google Colab will fix this bug. I think they have already put out a notice that they will patch this bug in the time to come. Okay, coming back to our notebook. First things first is we need to have our OpenAI API keys. Again, as I mentioned, these are keys which allow you to access the ChatGPT models in a programmatic way. And to get these keys, we need to go to the OpenAI website. And if you go to this OpenAI website here, you can go to this tab called API keys and then just copy paste it or save it on your notebook or on a notepad or wherever you feel safe. Just a quick disclaimer: you would need a credit card to use the OpenAI API keys, so that you have to pay as per as you use. But these costs are absolutely minimal. As I said, to process around 750 words, it roughly takes around 12 paisa Indian rupees or $0.0015. Okay, so let's begin. So first things first is we'll need to add our OpenAI API keys. So I will just run this, and it will ask me to, enter your OpenAI API key. Now I have already saved it on my own notepad somewhere. So I'll just copy paste the keys, and then now the keys are added. Uh, so okay, now we'll start working with the Jupyter AI Copilot. So I will first load this Copilot library, which is called Jupyter AI. And so now this extension is added. So, so now how do you work with this Copilot? So as I mentioned, pair programming is a workflow where you're working together with AI. Uh, and here the AI is the Jupyter Copilot, which is powered by ChatGPT 3.5. So to use these Copilot functionality, what we use is something called a magic command. So this is the simple syntax of how to use this magic command, which is like this percentage AI, then OpenAI ChatGPT 3.5 Turbo. So this is the model that is being used by this Copilot functionality. So for example, after putting this magic command, I can ask it to, write a short poem about ChatGPT. So let me go and press it, and it's just processing, and it has come up with a very interesting poem, as you can see here. But definitely, when we are doing biomedical data analysis, we are not interested in text. We like to get the code as much as possible. We are really not interested in the text part. I mean, you can use the free version of ChatGPT, which is really verbose, which spits out a lot of text along with code. But when we are doing a biomedical data analysis using Python, using a Jupyter notebook, it's much better. The workflow becomes much easier if you're just sending in the instructions and you're just getting the code, executable code, and then you're executing it. To show you like a warm-up exercise, what it means. So what I have again done is I have added the magic command here, which is the Jupyter AI. Then this is the model that we are using. And here I have added some additional flags. F format, -F means format, and code. So I need the output of this command as a code only and not necessarily text. So I have given now a simple prompt. So generate a function to add two numbers, then print out the sum of 2 and 3. So let me go ahead and press this. And you can see the AI generated code has been inserted below. Again, it's a very simple function. But what is really interesting is you got the executable code directly in the Jupyter cell. So I can just go ahead and just execute it. And there you go, the answer. What about plots? Again, this is again quite easy. So again, I will show you. I have included the Jupyter. I have included the magic command here. Again, you can use -F of -- format code, which means that I only need the code as an output and nothing else. So here, my prompt is create a function that plots a circle of radius r. Let's plot it, and with a radius of 4. And there you go. You directly got the code in your Jupyter cell, an executable code, and I'm just going to execute it. And there you go, a plot of a circle. So that's in a nutshell of how pair programming works. You are working with AI together to analyze this data. So it really expedites your coding workflow. So now, before I actually start working on the data set, I wanted to provide you a few tips that will really help you to craft better prompts, craft better instructions, so how to work with this AI. So let me go back to my slide again. I hope you have already scanned this, but don't worry, you will also get the QR code at the end of the presentation. But let's talk a little bit about prompt engineering. Pair programming, I already mentioned. So when you are collaborating with AI to write code, it's actually called pair programming. So prompt engineering, to briefly explain what it means, is essentially creating better prompts, to get the generative AI models to produce very precise outputs. And some simple tips that you can use to get better code out of the model, better quality code out of the model is keep the prompts limited to one task. Do not ask in a single prompt to do multiple different analysis. It's always better to do one analysis in one prompt. Uh, Please define the inputs or the data set in the data analysis task in your prompt. For example, you should define it like you are given a data frame whose columns are genes, gene expression value, or so forth and so on. So please try to define as much as possible what the inputs to the data analysis task are. Again, you also have to indicate clearly what the output should be, whether you need a number as an output or you need a plot as an output. And it's also important, I mean, it's also a recommendation that you should wrap your code in a Python function. That is, you should instruct the model to return a Python function instead of just code. The reason being that Python functions are, then you can easily reuse these functions later on for some other task, and these functions can also be saved and used as a standalone functions later on, and it becomes much easier to share with your colleagues as well. So it's always better to get out Python functions than simply getting a couple of lines of code at one point. And finally, it's very important that we would like to emphasize that AI it kind of behaves like a human. It does not get things correctly in the first go. So be prepared to iterate, be prepared to fix some of the bugs sometimes yourself. And that is why the knowledge of coding is quite important. Actually, you simply cannot outsource all the time. But of course, it will write the code, but you should be cognizant enough to spot certain bugs in the code. Or sometimes you have to rerun the prompt so that it can create a better quality code for you. So which means you will never get a code. In most of the time, getting the perfect code is difficult. So now, again, let's now, with these instructions now, we'll now jump back to our data analysis task. Let's go. We are going back to our notebook now. All right, so we'll first load this data set, which is something called a workshop data.csv. And I will just load and give the head. Maybe Alok, you can quickly
Sure.
give a rundown on what is, just 1 minute, just
Sure.
let
Yeah.
me know what.
So guys, here over the index like 7316-101 and all, these basically represent the sample IDs. Okay. So they are the identifier for the sample IDs of the patients. Then over the columns, we can see like ENT, PD4 and all. These basically represent the name of the genes. Right. So this is HUGO nomenclature of the genes. Then over the last column, you can see is basically clinical status. So clinical status means basically the patient 7316101 belongs to which particular class. And as I told you, we have included only GNG and MBL. So we will have only these two classes in our data. Then as I told you that the data is normalized. So the data is log normalized and Z score transformed. Okay, what is all these things we will discuss in detail some other time. This data is log normalized and Z score transformed. Therefore, we can see some negative values as well in between the data. So this is the description of the data and how our normalized gene expression data looks like. Yeah, next, Neil.
Okay, so the first analysis now will perform, which is how to check the differential express change using t-test between the two clinical conditions. So now to do this analysis, we have already crafted the prompt here. So first I add my magic command, indicating I use this ChatGPT 3.5 model and I need the output only as code. So now here is the actual prompt that we have used for this particular analysis. You are a bioinformatician with experience with Python programming language. You are given a gene expression data where each, I think, each row is a
Each row is
each row where each row is a sample and each column is a gene expression value. So we are trying to define as much as possible what the inputs are. The last column, which you can see here, the clinical status, is a categorical variable with classes G and G, and I think there is also one more called MBL, which are the two different types of brain cancer, I think, brain tumor. Now, this is the function. This is the prompt. Design a function to perform t-test between these two classes for each gene, and return the results as a data frame with t-statistic and p-value. Again, we have indicated in what format we need the output that should be generated. So now let's go ahead and press this and see what happens. Okay, so now we have received some results, and you can see it has given a particular code. Now I don't need to reload this data set again. It is sort of, it has included that the data should be loaded, but we have already loaded the data set here. So I don't need to do this again. So I'll just modify this, I'll just put it here, and I just don't need this stuff. And let's see if I can print it. Okay, we have an interesting error. The data frame has no, okay. I think sometimes it happens, as I said, you never get the perfect code in the first go. But let's see if we can try to debug this. So t-test is,
Neil, can you show the error? Like, what is the error exact?
yeah, it has taken the data frame has no.
Great.
Yeah. I think let's see if we can correct this error in
real time.
No. But I also like try to generate this code earlier, but let's see if there was something else in the previously when we generated this. So there is also a notebook called Final where we tested all these functionalities out, and that time we try to debug, all these. So here, I think what's the difference here? So the genes, let me see. Genes are the t-test. Consider the t-test as a data frame. Okay. Okay, for gene in gene. So it has taken a t-statistic and p-value, and then so because I have already a working code previously, what I'm going to do is I'm going to simply copy paste this code right now for the purpose of this demonstration. I mean, I think when we started working on this problem, we saw a similar error. So
Yes.
to be mindful of time, let's just copy. I have copied the code, and let's see if this gives me the correct answer. Okay, good. So this is the same code which was generated previously when it was working, but now again we need to like debug. But now this works okay, it seems. So here we can see for each gene now we have the t statistic as well as the p value being calculated. Now, one of the shortcomings of this method is that whenever you're performing an analysis, you need to like sort of redefine everything. You need to formulate the whole problem from beginning. For example, if I ask it to update the previous function to also calculate the log FC values, which is fold change calculation values, it will not work for sure. So let me show you. Let me go ahead and press it. It has given you some code. So let me see if this works.
Yeah, but it has not updated the code, right?
It has not updated the code. So we asked
Right.
this thing to update this particular function, but this isn't what we wanted. So which means that every time we want to do an analysis, we have to formulate the whole problem in its entirety. So now let's see differentially expressed genes with t-test and log fold change calculation. So let's hope that we get the code this time correctly. So again, here the prompt was, you are a bioinformatician having experience with Python programming language. But this time we also ask it to design a function to perform t-test and the log fold change calculation between the two classes. So let me see if I can get this. Okay, I have a, some value. So it has given me both these answers.
But again, it will give the
Uh,
error. Again, it will give error
why did
because again it has used the append function again, Neil. And data frame do not
okay.
have the append function, right?
Yeah.
Yes. But we can try and
So
show.
I think
We can try and show the error.
yeah. This is Ritasan. Yeah, you're right. Uh, but I can already try and show this. This will not work for sure
Absolutely, it will give
because
error. So,
oh, data is not
no, sorry,
defined by
you need to pass the
EXP.
data as well. Gene exp, gene exp, and C. Yeah,
Okay. So what I think if we have the same error, I think data frame does not have the
append function.
append function, but we also got the same error previously when we were doing. So I will show you how we tested this, corrected this code previously. So this is the again same, same thing we tried out last time. We also got the same error. So how we corrected this was we made a small change. Instead of data frame, we used a list variable. Uh, we actually used a list, and we did all the calculation appended in a list, and later converted this list into a data frame. So I will just copy paste this whole code again and go back to here. Okay,
Great.
so now you can see the log fold change calculations here being done.
Yeah,
So we made a
yeah. Neil,
small,
can I add
yes,
something here? Yeah, can I add something here?
yes,
Sure.
yes, please.
So guys, here you saw one thing that like by using ChatGPT or any AI-based tool, you will definitely get some error. It is not really going to return you the best answer, as Neil said in the prior as well. Okay. So therefore, like you need to have some programming understanding as well, right, Neil? To figure
Yeah,
out the code,
exactly.
like where you are getting the error, why are you getting the error, and how you can resolve the errors as well. Okay. So yeah,
Hmm.
next. So yes, like now
Okay,
after
now,
having the yeah, so, sorry, go ahead.
no, you can explain this volcano plot.
Sure. Great. So, like guys, you can see here the what we have got. So we have got the gene, T statistic, P value, and the LFC. So let me explain you the meaning of this. Okay. So over the rows we have the gene like row index 0. We have the gene ENTPD4. We have the T statistic value, corresponding P value, and the LFC value. Okay. So let me explain you what is the meaning of this. Okay. So where we had the two different classes, GNG and the MBL, and we told the prompt. Neil, can we please check like the prompt we told MBL versus GNG or GNG versus MBL for the log fold change? Up, yeah, great. So with the classes, they just detail log fold change the class for gene. Okay, fine. So can we go down in the code? We can see in the code as well. Yeah. So it is doing. Sorry. No, no, in the code, Neil.
In the
The
code, you
later
mean you
code.
want to see? Okay.
Yeah, this code, yeah. Group 2 dot mean and group 2 is MBL, fine. So the code is basically performing here, comparing the MBL versus the GNG. Okay. So now guys, so
Hmm.
it is basically log fold change. What is fold change? Fold change means basically to what level one is expressed more in comparison to the other. Okay, like suppose that we have two values A and B. A is 8, B is 4. It means A is two times of A. Sorry, B is two times of A. Okay. And then when you're going to take the log of this, okay, so log of 2 will be 1 with respect to base 2, right? So we take the log because if the value of the fold change is above 1, it will be greater than 1. But if it is below 0 and 1, it will be a negative value. Okay, sorry. Yeah. So if the value is going to be greater than 1, okay, it will be greater than 0. And if it is between 0 and 1, it will be less than 0. That it will be a negative value. Okay. So for that reason, we take the log fold chain. So in our answer, like, can you please look at the answer, Neil? That is, yeah, fine. So here we can see that all the genes have got the negative value. It means that the uh, group 2 minus, yeah, fine. It means that MBL is down regulated in all these genes. Okay, so it means that these genes are down regulated or lowly expressed in MBL class. Okay, so this is the meaning of this. Then p value, like we are going to apply a threshold. And if the p value is less than 0.05, if we have decided threshold at 0.05, it means that the obtained result is significant at this particular cutoff. Okay, we will say that. So now we can see that the second value, ppm1e, it is having 0.012 or something, and log fold change this much. It means that this gene has a log fold change value of this much with a significant p value. Okay, so this gene is down regulated or lowly expressed in MBL class. Okay, yeah. So this is how we interpret this particular information. And now we will try to look at the volcano plot. That is how we can visualize this information, because we are not that good in reading the values, right? So we always try to imagine the things in the visual format. So for that reason, we are going to plot a volcano plot. Why volcano? Because it will look like volcano. Yeah. So now we can explain the prompt news.
Yeah, so again, this is a we ask it to like design a Python function, to build a volcano plot. Again, based on Alok, what you have already said, we indicated what the data frame is. So let's keep our fingers crossed and let's hope that this time the code comes out
So
perfectly. But
yeah,
no worries, we have already corrected some of this code.
absolutely.
Okay, let's see. It has given us PLT show. So I will just put this function here. So DF
Volcano
should be
plot.
t-test results, right?
Yes. So we need to give the
Yeah.
t-test result.
Let's hope. Okay.
No, no, no. It is giving wrong. Okay. Sorry. So let me check. Log fold change minus log 10 p value. So this is wrong. Can you look at the code, Neil? Can we look at the code? This is just reversed. I can understand it has not taken the log fold change, log value, I guess. Div df absolute
It has
log
taken the
fold.
absolute.
Yeah, that there it is correct. Colors
Yeah.
red. This what it has plotted. Where is the plot? PLD dot
This is the
scatter.
plot.
Yeah, it has not taken the log value.
Okay.
Minus log 10 p value. It is some error it is doing. Can you look at the code? No, sorry,
Okay.
sorry, sorry. Look at the code
So
in
we
the
need
code,
to.
yeah. So PLD dot scatter. Yeah, there it is the error name. So it is doing like -1 into DF value, but it should be minus log base, because we take the value with respect to negative log. Okay. So guys,
Okay.
here again comes the domain understanding, because I'm the person who do all the things. So I can feel that there is some error in the code. And the error is basically here that over the y-axis, can you look at the plot name? Sorry, yeah. Plot. Yeah. So you can see here on the y-axis, it is writing minus log 10 p value, but in the code, it has not taken the minus log 10. Right. So it has given this particular, for the labeling, it is correct, but the coding, it has not taken the log value. Yeah. So we can take the log value there, Neil, or for right now, we can just copy the code here.
Yeah, we can the code that we already previously worked here. So here,
Yes.
this is the corrected code.
Yeah,
So
like
here
here
you
we
can
see the,
see the log
yeah,
10 value. So this part was missing in the previous code. So
yes.
if I just copy this code,
Yeah.
and
Paste it here.
there you go.
Awesome. Great. So fine. So now guys, here if you're going to explain this curve, so let us see all these gray colored genes. These are what are down regulated. Sorry, these are neutral genes. They have not they're not differentially expressed. And what is the
Hmm.
differential expression condition that we mentioned in the prompt, right? So Neil, can
Hmm.
you look at the prompt so that the students are able to understand, right? Great. So you can see here that the differentially expressed genes, we have mentioned the condition log fold change greater than 1, and the p-value less than 0.05. Okay, so
Mhm.
any gene following this condition is going to be called as differentially expressed. If the value is going to be positive, yeah, down Neel, plot. Fine. If the value is positive, that is greater than 0, then the genes are red colored, and these are upregulated genes. And these blue colors are the ones of downregulated genes.
Mhm.
Okay. And the gray ones are basically not differentially expressed. So these are the set of genes of our of our
Mhm.
interest, right? Yeah. So now we can go
Okay.
to the next.
So yeah, finding differential express genes. You want to explain this?
No,
Alok,
no,
maybe
like,
we should
yeah, because we have
go ahead.
7 more minutes only
Yeah. So let's
for the
finish
coding
up
part.
this
Yeah.
PC analysis and then
We'll take the Q&A.
yeah, we'll do this PC analysis quickly and then try to wrap this up. Okay.
Yeah.
Okay. So here we finally we ask it to write a Python function, that takes a list of features and a data frame. I think PC analysis most of us are probably familiar with. So we have asked it to do we have instructed it to filter out the columns in the data frame using a list of features. Okay, okay. We should first now execute this, right?
Yeah.
Uh, differentiate. Okay, let's see if we can get this. Okay.
Yes, it has given correct result.
So can you explain this,
Yes,
what
so
we have
like you
done
can
here in
execute
this?
before I can explain there. Yeah, fine, guys.
Yeah, yeah.
So in the plot here, all the blue and the red colored dots, the genes
Yeah.
are of our interest, right? So for the further downstreaming task, we are going to take these genes and we will be trying to see in which sort of the pathways the genes are enriched in. And with the help of these genes only, we are going to perform the PCA analysis. Okay, so we'll try
Mhm.
to see what extent these genes have the potential to discriminate the
Mhm.
two classes in the PCA as well. Okay, so for that reason, this is just a function to filter out those genes. Okay, yeah, and you can see here these are the genes of our interest that we've got as a list. Great, yeah. So now we can go to the PCA.
Okay. So you can explain the PCA code
Sure.
if you want.
Yeah. So right here, guys, now you know that we have taken the gene expression data. We have also got the differentially expressed genes, right? So what is the task now? We need to filter out only those genes and the last column. And for the PCA, we will just like take all the features except the last column and perform the PCA upon that. And then we will add that last column and visualize the data. But right now, again, we are getting some error here. And let's see. So
Today has been a
the
bad day. Okay,
yeah.
I
So
think
again,
this
again,
is the
got this
same
error.
error we observed here
Yes,
last time.
right, right, right. It is giving the same errors again and again.
Okay. I think this is the same error. Mhm. So I will just copy paste this
Yeah.
code. I think the error was I completely forgot what the main issue was, but I think we corrected it. Let's see. I hope this works now.
Okay. And
Okay,
next we
so
go to
data,
data frame.
data. Okay, there you go.
Yeah, right. So yeah. So now guys, like here you can see that based upon those features, the GMG and the MBA classes are present in this format. And then we can see that there is some sort of not good cluster, but these MBL patients are present together, and the GMG patient, the blue colored patients are present together as a cluster, right? So
Mhm.
PCA help us to visualize our data in this manner, right? Yeah. So now Neil, we can also show the ML modeling just right because that is the last remaining. We can skip the
Okay.
enrichment and we can just show the ML modeling, right?
Yeah, yeah. So let's just show we were training like a simple logistic regression model to predict the two types of cancer which we have included in this data set. Again, let's see if it gets it correctly this time.
Yeah, guys. And again, in this, like you see that we mentioned all the ML based words here. Okay, that is like we want
Yeah.
what sort of the results, outcome, and all. So you need to have the domain understanding of machine learning as well here to perform these
Yeah.
kind of the tasks. Again, it is given error. Nice.
Today we have
So,
had a
so it
bad
is the
day.
same error
Oh
here.
yeah,
Like
this is again
including
a known
PC.
error.
Yes,
We know
that
this error
is
because
right. And the error basically, like those of you guys who have little bit any understanding of machine learning, you'll be able to understand the error. That is like this is encoding error. So we have not encoded
yeah.
the categorical values with the integer. So that's why it is giving the error right now here. Okay. So
So
now we will
so
figure out the
here
error here. Yes. Sorry. Yeah, you continue.
so here yeah we will just change the GNG and ML. So these we did not change in the processing. So now we just encoded it with 0 and 1, the two types of classes in this example. And let's hope and there you go
Awesome.
the receiver operating characteristics.
Right. But
Uh,
it's not printed
yeah.
the values.
Yeah, okay. So I can also add some print statements, right?
Yeah.
Let, let me just put it like this, quickly. Let me just put accuracy for timing and I can show you
Yeah.
the accuracy
Yeah.
value here.
So yes.
So as you saw, the it was a little bit tedious to work with this generative AI. Of course, it generates code. It gives you the full-fledged code. Now, keep you in mind that the scripts that were generated today, when we were testing it out, it also did not generate in the first go. We had to like iterate it. But often the fixes were very minor, like one liner here, one liner there, and then the whole code we were able to get it running. Now we are using the ChatGPT 3.5 model, which is also the free version model behind the ChatGPT web application. So this is not the most powerful model right now. In my experience, GPT-4 model is way powerful, and it tends to create much more accurate code than GPT 3.5. And but then using it also is a bit expensive, in my opinion. It's a bit expensive. So I'm not sure if we have time, but the basics of Git. So I also wanted to share with you what Git version control system is. So essentially, it can be used to track changes in your data analysis scripts, experiments, and so it's important that whenever you're making some changes in your code, these changes are documented, and then someone else could actually be able to reproduce it later. Uh, and then this also allows you to share your code via repositories like GitHub. And then you can share the repository with others for peer review or even include it in your publications as well. A typical workflow looks like this. You start from a main work branch, then you can like branch out from this main branch and create something called like a demo branch. So quickly show you like how this would work. So here I have created the repository, which is the on the main branch, which sort of contains this final work notebook, with all the instructions. But say I also want to add the notebook that we showed you during the live demo today, which of course it wasn't perfect at the same time. So there'll be some differences from the actual finalized notebook. So this is quite easy to do here. I'm using the UI here, so I can create a demo 13 Jan branch. So I'm creating a demo and I can add a file here. So I will, in this case,
The screen
uh,
is not visible, I guess. If you're, are you
oh,
presenting
okay.
something?
Share this app. Okay,
Yeah.
choose this app. Uh, so I can choose a file here. Uh, uh, so this is the demo workshop that we showed you. So I will just upload it this demo and say add demo workbook, and I'll just commit these changes. So now in this demo 13 branch, I have the finalized notebook, when we tested out. And also now I added so this is the final notebook and this is a demonstration notebook, which we use today to show you during the live demo. And if you go to the main branch, the main branch does not contain the demo notebook. So this is one of the ways how you can uh, track the changes in your code. Often we would make changes in the final this notebook itself. So there's also possibilities to track changes in a single file as well. So this is often used in bioinformatics and also almost in all levels of software development and data science. So this is a very important skill to learn as well. Uh, so going back to wrap this presentation up, we do not have enough time to talk about data confidentiality today. Just to quickly, one thing I have to tell you that the data that you work on, especially this bioinformatics data, this data does not belong to you. It belongs to the organization or the university. So be careful of how you are treating it, how you're treating
Yeah.
this data, So do not upload this data on your private storage or on your private or do not share this data with ChatGPT web application. So what the web app does is it uses whatever data you send, it uses this to update its own model, which can lead to like confidentiality issues. Whereas using the API, which we used in this workshop, so none of your actual data set was sent to the ChatGPT models. And when you use this API approach, which means you're programmatically calling the ChatGPT models, this does not use your data to improve your model. So this is a much slightly safer way than directly working with the web application. And finally, as I said, learning generative AI tools is not enough. We encountered a lot of issues, but a lot of these issues, it was uh, using just a single line of code, we were able to correct it when we were testing it out, testing this same notebook. A lot of the code was actually being generated in a very similar fashion, and we were able to spot these issues. So what I want to emphasize is that, of course, using these tools will really accelerate your data analysis task, but you also have to have, of course, a very good understanding of how biostatistics work, how machine learning works. You need to have some domain knowledge, and you should also have knowledge of uh, the basics of programming so that you can spot these errors and correct them. And which means you should be able to critically access the output of your generative AI tools. Finally, you should take care of how to conduct reproducible research and take steps to manage your data and code confidentiality. With this, I think we are at the end. Apologies that we went a little bit over the time today, and also apologies for the hiccups in the demonstration. It's very natural. We expected that this would happen, and that is why we had already prepared this code previously.
Okay.
And yeah, Alok, you want to say the final words?
Yeah, thanks. So we'll take just 2 more minutes, guys, and then we'll go to the Q&A session. So basically, like
Yeah.
as Neil said, that in order to use generative AI and all those things, we really need to have good fundamentals understanding, right? And for
Mhm.
that reason, we have started this particular company where we are basically trying to teach the student the fundamental level of the things, where we teach biostats, machine learning, programming in Python. Or Neil, next slide. Sorry. Yeah, great. So we have a large number of the courses like biostats with generative AI, clinical data science, which will be a bigger course, biological data analysis, NLP for biomedical data, CV for biomedical data, then Python and R. And just to look at the expertise of our instructors, so all these people belongs to the industries or academia, right? So we do not, yeah, fine. So like we have Neel from Roche. So we and Neel are basically collaborating together. Okay, so he will be basically leading the complete data science part of our company. So we'll be looking at something. So then we have Vasu from Mercedes, and we have Dr. Hara, who is a medical, who is a doctor pursuing his MBA, MD from UCMS, University of Delhi, and others. Okay, so if you are a beginner and want to understand the fundamental of all these things, okay, then TechMedBuddy or Brass is one of the right place. We are not doing our marketing, but we're just telling that we are someone who works at the fundamental levels. Yeah, next slide, Neil. And also, like you can, I have shared the link, so many links here in the chat here, where you can follow us over YouTube and the link, YouTube, LinkedIn, and all to get the coming updates. Okay, and yes, we know that like this session was a little bit fast today due to the time constraint. So this video will be uploaded over the YouTube, and then you can just play with the speed and have the session again and again to get the things. Also, the code file is going to be available by the open repository. So, and these are our LinkedIn profiles where you can just scan and follow us over LinkedIn and ask any questions that you have in mind. Yeah. So now we are officially done from our side. So feel free to ask any doubt, anything that you have in mind. Yeah. Thank you so much, guys, for joining and having being with us for so long. Yeah.